Good afternoon. I'm very glad to be here with you and we'll try to give you a message of the importance of, at this stage and in the consortium, of extending and internationalizing the experience that you have accumulated, not only in the research field, but also as families. As my background, I've been a president of the Latin American Society for Glycobiology. So in the last 10 years, we have made efforts to make glycoscientists in Latin America know each other and also try to work in different fields, including CDGs. Every 2 years, we held a meeting of glycobiology in Mexico where we gather, and every year, or every meeting we talk about CDGs, among other topics in glycosciences. Also, in the last 4 years, we have been funded to raise a glycoscience in health network in Mexico. And maybe you don't know, of course, you know CDGs and the importance of glycobiology there, but you could take the 10 most important diseases: diabetes, obesity, Parkinson's, Alzheimer's, and many cancers. And in all of these diseases, glycobiology and glycans, the same things that are not working in CDGs, don't work in these diseases also, and are fundamental to know these diseases and also to develop new solutions. So, instead of only working with rare diseases, we want to join efforts and try to advance glycoscience to better understand how glycans affect disease, CDGs, and other diseases that sometimes can be seen as more important by the authorities, although we know that rare diseases, even if they affect low numbers, are also very important for society. So, well, just so you know, here, I don't know if well, it doesn't seem to appear there, but in yellow, there's the state of Morelos. Our lab is south from Mexico City. Oh, okay, yeah, thank you. So we're there in Cuernavaca, south from Mexico City, 30-minute drive from Mexico City, and it's 3-hour flight to Tijuana. That's the flight I took when I came here. And it's a very nice town, colonial town, and it's also the birthplace and where he was killed, one of the revolutionary leaders in Mexico that maybe you know or you have seen, Zapata, Emiliano Zapata. Maybe in the United States you know more Pancho Villa, but in the South, the other revolutionary leader was Emiliano Zapata. And so it's a very nice town, and Mexico is very centralized. So a lot of the hospitals and research hubs are mostly found in 3 cities. One of these cities is Mexico City, of course, a city with around 25 million people. And another one is Guadalajara over here. And other that we don't see here is Monterrey. So these are 3 regional hubs where sometimes it's good to have these centralized hubs because particularly to treat rare diseases, we can focus our efforts in these 3 main cities and try to bring changes in how rare diseases are diagnosed. So of course it is very important for me to let you know that Latin America is composed of 33 countries with a population of 626 million people. So this is approximately the double, I think, of the United States. And also importantly is that the genetical background, the ethnicity, makes it one of the most diverse regions in the world. One of the problems, not only dealing with CDGs, but rare diseases in general, is the lack of national plans, specific health initiatives or policies to attend rare diseases. And if you want to look for specific policies for CDGs, you will not be able to find them. And in those countries where there are plans to attend rare diseases. The problem is that the legal frameworks and the funding to make diagnosis, for example, or research are not present. Another important thing is that 30% of the population don't have access to healthcare for economic reasons, and 21% do not seek care because of geographical barriers. So one of the messages I would like to leave you so we can start thinking about how can we change this in a new stage for CDG research is that we should know what is happening in these 33 countries regarding CDG diagnosis, research, and treatment, mainly for two purposes. One should be to know what is the registry of CDGs, how many cases there are, how many laboratories are working in this. Maybe first from a perspective of human solidarity and solidarity with other families and patients that are suffering from the same thing. And that I can assure you that if we don't do nothing, very probably most of them will live and die without knowing that they had a CDG. So I think from the human perspective, that is what we must be driven through. On the second hand, it's not only solidarity, but also profiting of it mutually. Why? Well, through the hundreds of patients that we could find and that are already present in Latin America, we could extend our knowledge of the impact of different mutations, even new mutations, and understand better how these diseases progress, and even try to understand new solutions to the same diseases that are found in the United States and in Europe. Also, this would significantly increase the number of patients that could be registered to clinical trials. It is important to know that even though there are difficult social and economic situations in Latin America, there is enough infrastructure and hospitals in many of these countries to perform adequate clinical trials. That all this information would be also information for you. And as you know, CDGs do not respect boundaries and nationalities. And the more information and biological data we have incorporated into our registries, clinical and in the research type, we could, well, empower ourselves to make better decisions that could impact also your children and your families. And of course, it may sound strange, but I will not be giving you precise information of what is happening in Latin America, because we don't know what is happening in Latin America in a precise manner. But I will give you, I hope, some information that may shed some light about the challenges we must face and how we could do to improve this situation. So in this slide, you have 8 countries in Latin America: Argentina, Brazil, Chile, Colombia, Guatemala, Mexico, Uruguay, and Venezuela. Maybe you have heard of them, maybe even you have traveled to these countries at some point. All of these countries have reports of children affected with CDGs. In parentheses, the numbers on the side of each of the countries' names indicates the number of published articles with participating laboratories in these countries that have published in the area of CDGs. So why is this important? Because maybe if we really want to change things and don't stop missing opportunities, as I think we have, is that we must think that we have 8 countries in Latin America where there are already laboratories that have been sensitized to working with CDGs, and that with correct motivation and structured and coordinated programs, we could start making these laboratories national referral centers for CDGs. And particularly those countries like, for example, Argentina or Brazil that have published already a lot in CDGs could also be technological platforms where other countries could send samples for more complex studies. These countries represent 78% of the Latin American population. So approximately, we still have two countries with 200 million people where we don't have seen any reports of CDGs being diagnosed. And there is no reason to think that in these countries there are no people affected with CDGs. So what we must understand is that we have some countries where they are diagnosing and researching CDGs. That doesn't mean that all people with CDG are being diagnosed. So these countries do CDG diagnosis, but very certainly there are many people that don't have access to the healthcare systems in those countries. And another part of the countries, 200 million people, where there is nothing being done probably regarding diagnosis and, of course, not research. So I think we have a lot of kids, a lot of families that we can help if we start thinking how small steps could we take as families and researchers to try and make more diagnoses in Latin America. So I will talk to you a little about what has been made in Argentina and in Mexico. And I've put together these two countries because since many years ago, we, with Carla Astellano, who is a researcher over there in Argentina, always have talked and made some ideas and contributions to improve CDG diagnosis in Latin America. In 2010, we made the first Latin American symposium on congenital disorders of glycosylation. We had a lot of researchers there. If I remember well, Mark Patterson was there, Hudson Freeze was there, Kimillo Raymond from the Mayo Clinic was there. And then we repeated this symposium in 2014. So it has been some years ago now that we haven't been able to do another symposium. This is not because we are not interested in, but as already a little bit our colleague from Portugal, that is not in Latin America, but in Europe, well, funding is scarce. And so maybe an important information for you is that we have to understand that in most countries of Latin America, the percentage of expenditure of the GDP in research is 6 to 10 times less than is made in the United States. And you can also imagine that most of this scarce funding for research is directed to diseases that involve a lot of people. So rare diseases won't be adequately funded. Additionally, we have wrote some chapters in books. This was a recent chapter in a book that involves genetic diseases or metabolic diseases in children in 2017. And the importance to say this to you is that we also must understand that a lot or most of the people that could have a patient with CDG, even physicians, won't be able to read the material regarding CDGs that mainly you will find in English. So sometimes a little bit of initiatives regarding translating the material and the knowledge that you already have in Spanish could, I think, really improve the life and the decisions that many families and even physicians could take. In May of this year, we will be publishing a chapter devoted to CDGs from a book series called Glycoscience in Health. And the audience of these books is dedicated to physicians and laboratories in Latin America. And so in this chapter, there will be a description of the biochemical features of these CDGs, but also the clinical information that is important for physicians to identify CDGs. This will be in Spanish and will be part of a campaign we'll be launching to give this chapter and additional information throughout Latin America, giving this information particularly to geneticists and researchers that are working with glycobiology and also specifically with CDGs. So in Argentina, Carla Astellano couldn't come. She says hello from Córdoba, Argentina. Córdoba, Argentina is here in the northern part of Argentina. And there she works in a center for the study of inborn errors of metabolisms, and also with the Córdoba National University. She is in a very adequate setting because in this center they attend children with metabolic diseases. So she could be a key ally in these efforts that we are doing to improve CDG diagnosis in Latin America. And there's the photo with Hudson and Carla and me last year in the 5th Latin American Glycobiology meeting we had over there, and where, of course, Hudson talked about the CDG research he has been doing. So in Argentina, this center is a national referral center for diagnosis and research of CDGs. And I will talk to you a little about the types of CDGs she has dealt with there, and particularly some of the advances they have done with some types of disorders. I don't know here in the United States how much of these types of CDG are part of CDG care, but I hope that this would be interesting for you. So as it happens in many parts of the world, there are specific diagnostic flowcharts that have been already established in the laboratory at this referral center. So there is a structured and systematic approach already in place to make the genetic diagnosis and, if possible, treatments for these patients. And these protocols are also followed by my laboratory in Mexico. And as you may know, well, it all depends on starting to use some biomarkers like transferrin to make that first biochemical suspicion of a CDG, and then proceed afterwards to enzymatic analysis, spectrometry analysis, or genetic studies. I think one of the important things is that I think that we could establish in many of these countries where CDG diagnosis is not being done, at least the test of isoelectric focusing of transferrin. That, if we make that little step, we can make a big contribution to CDG diagnosis. Afterwards, there will be problems to have access to more complex studies like mass spectrometry or genetic studies, and there is where some regional referral centers or directly with the CDG hubs in the United States, in Europe, or as a consortium, we could try to help out and identify crucial information to have a specific diagnosis. Particularly over there in Argentina, they are very interested, and they have a specific program for skeletal O-glycosylation disorders. So they have already established protocols where, depending on the imaging abnormalities that you can find in patients, they relate that to genotypes or genes that have to be sequenced in order to identify potential mutations. And here on the lower part, we have a specific pathology that is characterized by cartilaginous tumors, bone shortening, and deviation of bones that are related to mutations in two genes called EXT1 and EXT2. This has as a consequence a disease that is called multiple osteochondromatosis. In this disease, there are bone growths that are abnormal, particularly in the long bones, in the terminal part of the long bones. There will be growth of bone that is accompanied by pain. And normally, if the surgeons or pediatricians are not aware that these are genetic diseases, they can take out these parts of bone, and with two consequences. One, that they will not do the genetic studies and maybe won't find these types of abnormalities in other parts of the body, even in the pelvis, where, for example, the symptom may not be pain, but only constipation, and you will have a bone of 10 or 15 centimeters of diameter inside the pelvis. But also that if surgeons are not trained to deal at the moment of excising these bones, they have a big tendency to bleeding. So if they are not prepared, the patients could die in the surgery if they are not correctly managed for the hemorrhage that accompanies the removal of these bones. This growth of bones, as you can see here, or in the images, not only brings pain and impedes movement, but approximately 1 to 5% of these patients can develop in those sites osteosarcomas, so cancer, okay? And so that will bring a mortality, a very important mortality for these patients if those bone growths are not removed. So this requires adequate monitoring and interdisciplinary team to deal with these patients. And in Argentina, they have a very good experience spanning at least 10 years with these types of patients. The registry of patients they have with this disease in Argentina are 92 patients. So this, I think, is a good group where an initiative that Carla is promoting to create a first family organization over there for CDG families. Also, and this is important because it was funded by the government of Argentina, they will install a program this year of neuromuscular disorders. So they have a protocol also in place to identify different types of muscle disorders. And of course, inside these protocols are those diseases that affect O-glycosylation disorders. We have heard a lot of talks of these alpha-dystroglycanopathies, and also to determine the genetic defects that cause these types of CDG that affect muscles. So this is important because, as I said, not many countries have the willingness or the money or the vision to put money into CDGs, but at least this year they were willing to put money inside a big program for neuromuscular disorders. So this is the registry in Argentina. So they have 10 PMM2 patients, 2 MPI patients, ALK1 and ALK2, ALK13, 2 patients with COG1, and 5 CDG type 2X that are being studied to determine what is the gene affected. And in the O-glycosylation disorders, they have the beta-3 GALTL CDG that causes a syndrome called Peter Plus syndrome, short stature, and abnormalities of the eye. One patient, as I said, 92 patients with the multiple osteochondromatosis disease, and 1 patient with a dystroglycanopathy, the large CDG that causes Walker-Warburg syndrome. And there's also a publication, relatively recent, where it describes the 10 years experience of screening for CDG in Argentina. And as I said, Carla has the intention of founding the first Latin CDG Foundation to attend professionals and families, diagnosis, research awareness, to engage Latin American efforts to provide CDG diagnosis, research, and therapy, and also to develop a network worldwide to help patients and researchers that are interested in CDG. And one important thing is that we don't want to start everything again, because there's already experience from your part. So one of the things is how we can do this quickly and trying to have good results in the short term. The collaborations of Carla are in Spain with Belén Pérez, Pablo Lapucina, Daniel Greenberg, Susana Balcells, in Barcelona, in Madrid, with United States, Kimillo Raymond and Hudson, in Italy with Luisa Sturiale, and in Belgium also with Dr. Jack Jecken, that probably you all know. In my case, I'm in a different setting, so I am not in a hospital, and that could be not very advantageous for me. Nonetheless, I was interested in diagnosing CDGs when I came back to Mexico 15 years ago. And so in my lab, I do glycobiology of T cells. I also study how viral infection changes the glycosylation of infected cells. But as I said, I am very interested in doing the CDG screening and research in my country. This is because in 2005, we published during my doctoral studies in France, the first CDG that involved mutations in the Golgi CMP sialic acid transporter. So there is no sialic acid in the Golgi for all the sialylation, so glycoproteins don't have sialic acid, and this causes diseases that involve particularly a lack of production of healthy platelets and hemorrhages that can affect these patients. So that I worked in my PhD, so of course when I got back to Mexico, I said, well, I want to do CDG diagnosis over there because there hadn't been until 2014, the first diagnosis of CDGs in Mexico. So this is an important story because it's something that maybe we should have to do in many of these 33 countries that I said compose Latin America and which no CDG screening is being done. So we had some training with Dr. Hudson Fries and Bobby in 2010. They provided us positive control samples to correctly identify type 1 and type 2 patients. And importantly, we produced anti-transferrin antibody production in rabbits in our laboratory. Why? This is very, very important, okay? Why is this important? So the first step to improve CDG screening is for the laboratories to have an isoelectric focusing equipment. That's not very costly. We could give them to them. We could buy the 33 equipments for every country in Latin America. It wouldn't cost a lot. The problem is that every time you do an isoelectric focusing test, you need to use an antibody to detect the transferrin and the different profiles to identify defects of type 1 or type 2 glycosylation. If you buy it commercially, the costs go very high, and it's almost impossible to sustain in countries in Latin America. So what do? You produce the antibody using rabbits. You inoculate rabbits with transferrin. The rabbit produces the antibodies, and you use the serum of the rabbits as if it were a commercial antibody. The only thing, it's simple, but the only thing is that you have to do it, and you have to know how to do it, and this is very important. Not only to give the equipments of isoelectric focusing, but also give laboratories enough antibody so they can do the tests. So this is one of the things we can do, and we have trained some teams in Latin America to produce the antibody, and we have also sent antibody to other countries. We bought the isoelectric focusing equipment, and one important thing is that we have performed 539 tests until now, and all of them have been free of charge. So we don't charge anything to patients for doing these tests. Why? Because, again, in a context of poverty and difficulties, we don't want any barriers between the patient and a first diagnostic test. So we have done these tests, and most of the funding for these types of studies has been more personal than from a specific funding for CDGs. In Mexico, we have 3 patients that have been diagnosed with this type of disease, ATP6V0A2 CDG, and we have 3 type CDG1X also being studied. That our millennial student, I always hope he's studying CDGs in his phone, is trying to understand what are the mutations causing this CDG1X. And so with these 3 patients, 2 of them, we already published the results in 2014 and were the first cases of CDGs. And one important thing is that if you go to a country and you start talking about CDGs, but there are no diagnoses of CDGs, physicians won't really be interested. Okay, so making that first national publication of CDGs starts physicians and particularly geneticists thinking when they see a patient that it could have a CDG, and of course that they know that they can send him to somebody in the country, okay? So it is important in these 33 countries of Latin America to do a first publication of CDG. And so the 2 patients we published are 2 non-related patients with cutis laxa syndrome, so a syndrome of wrinkly skin, referred by a geneticist in a public hospital in Aguascalientes. That's a little bit of north of Mexico City in the central region of Mexico. So he sent us the serum over there to our laboratory. We tested it, and I'll want to talk to you a little bit about that. So these are the 2 patients we have. So this is the same patient. This is early in his life and a couple of years after. And this is important because you can see the abnormal fat distribution in the gluteal area, also the small short finger, the flexion, hyperflexion that he can have, umbilical hernias, as a floppy skin, and as you can see here in this patient, a very wrinkly skin that you can just poke like that. And one important thing additionally to the hernias is that he also had a cardiac defect. And one of the things that happens is that this kid, after a few years that we saw him, the wrinkleness was not as apparent as in the initial stages of his life. This, as in many CDGs, alerts us that we have to diagnose these patients very early, because sometimes the more visible signs of a CDG or a syndrome could disappear and make diagnosis more difficult. So we did the isoelectric focusing, we detected the abnormality. We sent the serum to the Mayo Clinic with Camillo Raymond that did the spectrometric analysis and confirmed the problems in the transferrin and also in another protein called apolipoprotein C3. So both proteins were affected. N-glycosylation and O-glycosylation is affected. So it's a mixed type of CDG. And this, along with the cutis laxa syndrome, pointed us out to a potential defect in a reported proton pump in the Golgi. So this proton pump is necessary to establish an adequate concentration of protons to establish the pH required for glycosyltransferase activity and protein secretion. In Mexico, we did the sequencing and we established 2 mutations in these patients. This is the first patient and the second patient. These mutations had already been reported in the literature. So one of them cuts the protein very early on, the other almost at the end. We published this report in Molecular Genetics and Metabolism Reports. And currently, we are doing genetic testing in their hometowns to determine the presence of this mutation in other members of the family and also in the vicinity of their local habitats. So one of the things that we are currently doing, and I think this is very important in many of the CDGs, and it's not being done as I think it should, is that we are doing some transcriptomic analysis of these patients. What does this mean? So most of the times when we want to understand a CDG, we know that the disease is caused by a specific mutation. And we understand the disease through that mutation and through that gene that is affected. But what we must understand is that when one gene is affected, other genes also change their expression. And so even though the mutation is in a single gene, the fluctuation in the expression levels of other genes also may explain why the patients are sick. So we are doing this analysis to answer 3 questions. Does a mutation in this gene affect the expression of other genes? Could altered expression of other genes explain the disease? And could we identify genes that allow mutated cells to survive and explore therapeutic potentials? So when cells have this mutation, they change the expression of their genes also to survive. So if we identify what genes that are normally healthy are allowing those cells to survive, and we can potentiate that effect, then it could be also an alternative to develop new therapies. These are the families of one of our patients, and this is the second one, recent photographs of both of them that they are currently well. One of the families could have come, but we didn't have time for the visa, but we hope that in the next event of CDG, we could have them here with you so they can also meet them and start internationalizing the network. Our collaborators, of course, have been in Argentina, Carla Astellano, in the United States, Hudson and Bobby, and Quimillo, of course. And importantly, in Mexico, we started recently a collaboration with Carmen Alaez of the National Institute of Genomic Medicine that will allow us to do exome sequencing in these patients. And this is a great contribution because we would easily sequence the genomes of these patients. Lastly, I will talk to you about Brazil. There is a recent publication of a 10-year experience in Brazil on CDG diagnosis in a specific laboratory of airborne errors of metabolisms of the Hospital de Clínicas de Porto Alegre in Rio Grande, Brazil. So they do over there the isoelectric focusing of transferrin. They have screened from 2008 to 2017 1,546 individuals. And, well, to make the story short, they were able to identify 4 patients. So it's always a big work of screening to identify a small population, but it can be done. 2 PMM and 2 CDG patients, 1 SLC35A2 patient, and 1 MPDU1 CDG patient. This is what is reported in this article. Nonetheless, we have reports also available in the literature with Brazilian laboratories participating, where they have done diagnosis of SSR3, SSR4, COG4, PIG-L, ALK6, and MAN1B1, and different congenital myopathies related to CDGs. How much time do I have? Is it over? Okay. So quickly, the technical challenges is to do clinical and lab awareness. There's underdiagnosis. We have to improve sample referral. We have to improve the isoelectric focusing of transferrin in these countries, to have analytical equipment available to do confirmatory tests, and also to have funding that in most cases we don't have specific funding to do this type of research. Social challenges, political and institutional awareness, the need for family organizations and networking, and public policy for rare diseases. The proposal for CDG Care is to pioneer networking, technical infrastructure, and advocacy for CDG in Latin America through a systematic and structured program. The accumulated experience that you have could more easily trigger successful clinical research and family experiences, and lead joint efforts to stimulate basic and clinical research that will benefit patients worldwide. And with that, I thank you for your time and patience, and I hope that you can understand that this, if we can put it forward, will help a lot of families that now haven't access to diagnosis. Thank you.